We are preparing for the SeekOneTM single-cell multiome ATAC plus RNA experiment. We are now ready to load reagents onto chip. Open the lid with the gasket attached. For chip S3, reagents must be loaded into specific rows, starting from row 1. Row 1 is for the reaction mix and cells, row 2 for beads, row 3 for oil, row 4 is for collection only. When working with multiple samples, load reagents across all chips by row, not one chip at a time. First, load row 1 for all chips. Then load row 2 across all chips. Finally, load row 3 with carrier oil. Do not finish all 3 rows on one chip before moving to the next chip. Make sure there is at least a 30 seconds interval between each row. The 30 seconds waiting time applies when loading a single sample. During multi-sample loading, this waiting period is naturally covered while loading across chips. Now the barcoded beads have equilibrated to room temperature. Vortex the beads for 30 seconds, spin briefly, and make sure there are no bubbles at the bottom of the tube. Add the fragmented nuclei suspension into the reaction mix to a total volume of 80 microliters. And gently mix. Dispense 78 microliters slowly into row 1. Keep the tip vertical, centered, and always submerged about 3 millimeters below the liquid surface. Let the tip follow the rising liquid to avoid bubbles. Wait at least 30 seconds. Aspirate 38 microliters of barcoded beads. After reaching the set volume, hold the tip in the liquid for 5 seconds before removing it. Insert the tip vertically into the center of row 2, slightly above the bottom. Dispense slowly while keeping the tip submerged and following the liquid level. Wait at least 30 seconds before loading row 3. Aspirate 120 microliters of carrier oil. Insert the tip tilted against the inner wall and dispense slowly. Repeat once for a total of 240 microliters. Now that all reagents are loaded, close the lid with the gasket attached. We're ready to run SeekOneTM. Tap open and wait for the tray to extend. Place the assembled holder onto the tray, keeping it level at all times. Tap close to return the tray. Select the correct program and tap OK to start the run. The run takes about 4.5 minutes.